Hi. Good evening, everybody. Can one of you please confirm if you can hear me? And then we'll start. Just give me a thumbs up or just maybe just say okay in the chat box if you can hear me. Yeah. Okay, great. Thank you so much. All right, everybody, welcome to the fourth live session of the course Cell Culture Technologies. So I can see familiar names. So some of you have already joined one of the previous sessions. I'll start by sharing the screen. All right, there you go. So I am Pallavi Gupta. I am a PhD student at IIT Delhi, and I am one of the TAs for this course, and I conduct these live sessions for you, which are mainly doubt clearing and to give you a quick recap of what was covered in the recorded lectures. So we have had 3 sessions so far. Um, Same Zoom link. The Zoom link stays the same, and the timing stays the same. So the main idea is that we'll start off with a quick discussion, or maybe not so quick discussion of what was covered. And of course, this is meant to be an interactive session. So if it's possible, I would like you to ask questions, whether it's through the chat facility, like just type it out, or you can unmute yourself and ask questions, even if they're hypothetical questions. Anything's fine. Okay, because now that you've joined this session, it'll be great if we can build on what things you know. And of course, there are not a lot of people joining, so it's okay. Like, just be as comfortable as you can. So we have that discussion for the first roughly 1 hour, and then we'll solve a few questions which would be similar to what you see in your assignment for that week. And then finally, we'll just take a look at the previous week's assignment. And if you have any questions in that, or if something's not clear or anything like that, and that's what we can discuss. So um, the session is being recorded, right? Yeah. And then this recording is going to be uploaded to YouTube, same time, like just after the class. And whatever material we discuss, which is primarily going to be this PPT, I'll upload it to the Google Drive, and you will have a link to all of this through the course page. So if any of you are joining us for the first time, I would love to know what you're doing currently and what has your course, like, even if you're not joining for the first time, I've been asking this question. So would any of you like to tell me what you're currently working, studying, and what's sort of your hope from this course, and how has it been so far, and what are you looking forward to, or what do you think, why did you take up this course? Just anything. And this would just help me to know if there's anything in particular that we can discuss that would help you. So I, for example, I'll start, and in the meantime, you can all gather some courage and tell me, or if you would like to start typing, you can already do that. So my work is at the intersection of cell culture and bioinformatics. That's computational analysis. So what I am doing is using stem cells. We talked briefly about what stem cells are in the first week. So they are pluripotent cells, which can become any sort of cell. So I am differentiating them into brain cells. And so I have a wild type set of stem cells, which are healthy, which have no mutations in that. And then I also have a knockout line, a knockout stem cell line, where a couple of genes are knocked out. And I just want to study what happens to those, what happens as a result of those genes when you look at brain cells. So these are neurons and astrocytes and glia. So what is the result? Because the gene product, so the RNA and the protein is only expressed in the brain. So there's no point as such in studying them in other cell types. So, and then once I grow neurons and astrocytes out of these stem cells, uh, I get RNA sequencing done for them, and then I analyze that data. Broadly, that's what the project is. So the first part is culturing stem cells, making these, differentiating them into other cells, and then doing a sequencing of that sample and then analyzing. So cell culture was relatively new for me. I've only been doing it for less than 2 years. So a lot of things that we are talking about in this course is also new for me. So we've had some discussions in the past, especially in the first couple of weeks, where uh, it's been new. So a lot of things were there which I also did not know, and then other participants of the course, they were able to answer it. So that's the whole point of this interactive sessions. So again, I would just like to take a pause, and if any of you would like to share what you're doing at the moment, and was if there's anything else you would like to tell us about. Just introduce yourself. That's going to be nice for everybody. And or if there are any suggestions or requests about the course or about these live sessions, because we are going to be halfway through today. So if there's anything in particular you would want me to keep in mind for the next 4 sessions, um, then I can work on that as well. So anybody? Yeah. Okay. So we have 9 students joining. That's great. So anybody? If not, then that's okay too. No problem. But I would really encourage you to be more interactive during the discussion of the next part, because otherwise it would just be very similar to me explaining the same things as were there in the recorded lectures. I would not be diverging too much from what was already there. So the point of these sessions is actually that you've had a week to look at the uh, uh, recorded lectures, and you have an assignment due in 2 days. So you would have watched the lectures, you might have taken a look at the assignment, and then if there's any problem, that's why we are joining here. But irrespective of that, um, we can start the discussion now if there's nothing anybody wants to say. That's okay. All right, cool. So we briefly talked about the sort of plates that people use, that is the base that people use where cell culture is done. So primarily, there are two materials we talk about. One is polystyrene, which is, think of it as plastic, and the second thing is glass. So polystyrene is generally to make plates, and we talked about plates that you can have petri dishes. We'll also call them plates. These are like 15 mm or whatever, 75 mm, based on their diameter. Uh, you can also have rectangular or cuboidal plates which generally have multiple wells in them. So these are named based on the type of uh, the number of wells that are there. We'll also have these different flasks where depending on the surface area is how they're named, T75, T30, and so on. Oh, hi. Hi, Rutika. Thank you. So all I was asking earlier was, would any of you like to introduce yourselves and tell me what it is that you're currently working in or studying, so that um, I have a better idea of who the audience is and what sort of things can I be discussing about. Because otherwise, this would just become very similar to the recorded lectures, and this is not a recorded lecture. So it will be great if it's more interactive. So if you've attended any of the previous sessions, or if you've watched through the recordings, or even if you've not, we still have, after today, we still have 4 more weeks of this course. So we'll have 4 more of these problem solving sessions. So is there something in particular you would want to be discussed here? Um, so yeah, mainly introduce yourself, and it'll be just great if the session is interactive. So keep asking questions. Oh, absolutely. You can unmute yourself. You can type. Either works. So whatever you. Yeah.
So, hello everyone.
Hi. Hi.
My name is Ritika Vise, and I am currently studying biotechnology. I am currently in MSc first SEM technically, and I completed my for BSc dissertation. I was in microbiology. So I want to learn about animal cell culture and wish to picked that up during my master's dissertation or for my future whatever studies.
Great. Thank you. That's really great. Thank you, Ritika. And Samriddhi, thank you so much. Yes, I remember. I remember your name. So, okay, great. Thank you. So yes. So what we've seen so far. So this is cell culture, but a theoretical course in cell culture. So it's more about giving you like a background on what to expect when you step into a when you know go into a project or a lab where cell culture is done. So because it's a very different set of things that you would be working with. Your pipettes stay the same, but then a lot of different things, like things are different as compared to a molecular biology lab. You're no longer in an open space. You have to be much more careful about contamination. And like the last week was entirely about the setup of the lab and what equipments are there and how things are arranged. So those things, while they may not be very evident to somebody who's dealing with cell culture for the first time, but then if you get a chance, so it may happen that, for example, like one of you said, you're currently doing masters, and let's say over the next 1 or 2 years, you gain some cell culture experience, and then for if you decide to do a PhD, you probably are going to step into a lab, and maybe that's a new lab. So this your supervisor is setting up the lab in a place. Okay, they're just starting off as a group leader. So in that case, you also have a say in where to keep what. So some theoretical knowledge is great, right? It's always helpful. So I, for example, have also like I was just telling you, I've done cell culture for about a year and a half, and again, I was also taught a lot of things, but these basics that I'm coming across in this course were also not. I was not aware of those, so I also learned them watching these videos. So there is like a 1% chance or maybe 5% that I may mess up somewhere. So for those of you who are already aware of this, if there's any of you here, uh, please feel free to stop me wherever I'm going wrong. I will try not to make mistakes, but that can happen. So so in the first week or the second week, we talked about what plates are used, different things. So broadly, it's going to be either plates or sometimes we will also be working with cover slips. This is not what we had covered. So the idea behind using cover slips is, so you might have seen cover slips in your, for example, in school. There's a very common experiment that people often do. That's the onion peel. You take out one layer of the onion, um, skin of onion, and you stain it with different things and look at it under the microscope. So in that case, you might have used a cover slip. Okay, the cover slip can be square, it can be rectangular, or it can be circular, as you can see here. And we just come to what's happening here. So now in cell culture, you are culturing cells, right? So, and we have been spending a lot of time with adherent cell cultures. So imagine if you have a well, okay, or a plate or a whatever it is where you are culturing cells. So now that's all covered because it's been taking, you are feeding it different food, media, growth factors, etc. It's there in the incubator. Now, when you want to image it, and we're not just talking about imaging it like we saw with the normal bright field inverted microscope. We are not just taking what you can see, just zooming in, but let's say you want to take some fluorescent images. Okay. So we haven't talked about fluorescent imaging right now, but for example, broadly what it is that you need to add a lot of things to it, to that well. Okay, you add antibodies, you add fixing agents, washing, you want to stain the nucleus, you want to stain specific things, primary antibody, secondary antibody, or there may be dyes that you add. So generally this thing is, it's going to take up some time. So maybe a day or two, depending on what cell it is. It may be a couple of days to do that entire procedure. So you add it, you keep it for 5 hours, you add something else, you keep it overnight, and so on. Okay, broadly. Now, those plates and those wells that we've been talking about, they're generally big, right? And you don't want to use a lot of your reagents on the entirety of that thing. Now, the thing is that um, when you have a well with a certain area, you want to fill it up to some point, right? So you will need to provide, when you're providing media, that's relatively cheaper as compared to what things you want to use to image those cells, right? So what people can do is, it's again, maybe if not, so what people do is you take that well and you don't just directly coat the well. You add these cover slips, these circular cover slips in the well. The well is also going to be circular. So you add these cover slips, will take up a smaller area than the well, and then you add your matrices, you're preparing the surface, that ECM you add on top of this culture, this cover slip, and then your cells are added to the well. So most of your cells are going to be on this cover slip. So then what you can do is, once your cells have grown and once you do a step called fixation, once you are basically killing the cell, but it's more like preserving the cells in whatever state they are. That's called fixing. So now you can take them out of the hood and now you can bring them to a normal bench where things can be done. So now what you need to do is just pour your whatever solutions there are to image those. Those things can be directly placed on top of this as a droplet. So a droplet can form like a um, dome on it, right? And then you're using less of the material. And then what happens is, the main idea, it's not just about saving reagents, but then you ultimately want to preserve, once you've made those fluorescent slides, you can keep them, ideally, you can keep them forever. You just store them at -20 and they're sit there. Okay. The only thing is you need this to be covered. So now think about it. You have a plate. So for example, imagine this is the plate and I have put a cover slip on top of this. And on top, or just imagine I have a screen guard on this. So imagine that is my cover slip. And now my cells are growing on top of this. And I'm just picking up that guard, that cover slip, and I'm putting them, as you can see here. Now I'm putting everything is on top of this. So cells are on top of this cover slip and all my reagents are on top. Now when I want to image it, I can't have this exposed to the surface because things are going to fall, right? What if somebody touches something there, right? What needs to be done is, and remember, we're talking about an inverted microscope. So, okay, inverted things comes later. What I want to do is I will invert this and put this on these slides. Okay. So there's going to be some sort of a media. It's called a mounting media. So I put a drop of that mounting media on this slide, and then I invert the cover slip facing downwards. So I have my slide, I have my cells in the middle, and then I have that cover slip. So my cells are protected and they're not squished because of that mounting media. And then I keep them to dry and then I can image them. Now, just an extra thing, when I want to image them, I'll again invert the whole setup because it's an inverted microscope. Generally, we have inverted microscopes and we talked about it in the last class. So if I can just draw this out. So it's going to be, you have this slide with you, right? And then I've got my cells growing here, or not growing, but my cells are here. And then on top of that, like my cells are on the cover slip, but that cover slip is on top of this. And now when I want to image them, so my objective is like this. This is my objective, and that's the stage. So what I'll have here is that I'm going to have my cover slip like this. And then I'm going to have my cells. And then finally, I have that slide. So my cells are anyway well protected, right? So nothing is going to happen to the cells. But the idea is I don't want to look at my cells through the slide because the slide is thick. Okay. And we've talked about a little bit last time that there's a only a certain range up to which your objectives can be far away from your sample. If it would have been in this orientation, the thickness of this slide will result in that your sample is now farther away from your objective. But now in this setup, your sample is only the cover slip distance away from the objective, right? So it's closer. So most likely it ensures that you will be able to visualize your sample nonetheless. Did this make sense or did I confuse you? If there's nothing, then I'll assume this is clear. But if it's not, please feel free to let me know and I can explain it again in a better way. Okay. So anyway, so yes, okay, great. Thank you. So, so the idea, so this entire thing was just to let you know that people can also grow cells directly on cover slips, because once your cells are grown on that, once they are adhered to a surface that you created for your plates, you can't lift it off, and you don't want to image the plate because it's a big setup. See, storing these slides is so much simpler, and you don't need to, when you're imaging, your microscopes are also already zooming in so much, so you don't need a lot of cells, you don't need a lot of area to tell you what's happening. A small cover slip is enough to tell you what's happening. So it's more economical, and it's definitely saving up a lot of space. You can just store this entire box. I think it saves, it can store like 100 slides. You can store 100 slides in that small box. So instead, your wells, however small the wells are, even if you're taking, so 96 well plates are not very often used for imaging because you will not be imaging everything together, right? This setup ensures that even if you have three things to image at one go, you're great. For 96 well plate, well, ideally you can have 96 samples in that, but very rarely do people have equipments to image all of that together, right? So you will have three wells fill in a 96 well plate, then another 96 well plate with five samples. So it's not going to be saving space or anything. Cover slips generally, yes, only in case of imaging, mostly. So, so in cell culture, there is a large part of planning that goes to tell you. So let's say I decide, okay, I want to grow um, XYZ type of cell. Like I said, I want to differentiate, suppose, my cells into neurons. I want to image them as well. And oftentimes you would want to image in triplicates if it's possible, so that you, if you want to do some sort of a quantitative measurement out of them, suppose the length of the neuron or the how much death is happening in a particular cell type, XYZ. Let's say there is some quantification you want to do. So generally it will be advisable to do in triplicates. And let's say I have three different types of cell lines that I'm working with. So I need 9 samples at least for imaging, right? But I don't want to just do imaging. Probably I want to do some RNA extractions also. Now, is one well enough for RNA extraction, or do I need 5 to extract enough amount of RNA? Do I also want to do something from proteomics? Do I also want to send it for something else, Do I also want to keep some wells extra just for a rainy day, if something happens? Now, generally, all of these protocols in cell culture, whatever sort of cells you're working with, more often than not, you will culture them for a month or two. So you have to plan today that, okay, 2 months from now, I am going to do XYZ things. For those XYZ things, I'm going to need N, M, and O number of wells. So that's a total of, let's say, 20 wells. So I need to prepare accordingly. Out of these 20 wells, I need 10 that should be cover slip based, and the other ones can be without cover slips. It's just better. Again, there are a lot of other things that people, I'm sure, do that I'm not aware of. But yes, cover slips more often than not is only for imaging. Otherwise, because it's an extra step, why do you want to add a cover slip in a well? The well is already a good material. So yeah, so that's that. Amongst the material of these two things, so the plates, they're polystyrene. Now, by itself, polystyrene is a so I am already recording this session, all right, and you will see, you'll have the recording. Just give me a second. Yes. Yeah. So the session is being recorded. So you'll you'll get the recording. So don't worry about it. Yeah. All right. So yeah. So the material of these plates by default is very, very hydrophobic. So nothing is going to attach to it as such. So some sort of a treatment is generally already done before it's given to the customers to introduce some level of hydrophilicity. And we'll come to more of these later. Broadly just speaking, so phobic means hating, fear, those things. Philicity is the opposite, so liking or loving. So hydrophobic means a surface that likes that doesn't like water, and hydrophilicity is what likes water. So just remember these things, that okay, polystyrene is basic, like it's somewhere in the middle. Glass, on the other hand, so glass is silicon, so it would have some. It also has some hydrophilic. It already has a hydrophilic group, so something, it's little more hydrophilic than polystyrene, but we still call it in the same zone for now. And this is one of the things that was discussed in the lecture, that when these, again, you have different quality of cover slips that come depending on, how expensive. Definitely, the more treatment is done, the more clean it is, and more things, uh, the more expensive it's going to be. So if it's possible for you to try out a few things, sort of, setting up or just preparing your slides or cover slips um, in the lab, then that does save on a lot of money, because you will use, like, for example, you can see this is one setup, and I'm not sure if you can see here, but there are something around like 15 to 18 cover slips that are used here, right? So you do use up a lot. So things like if you want to remove any traces of oil or grease in it, dip it in a soap solution. And etching is to just introduce more. It's, you don't want it to be smooth, you want it to have slight bumps. So that's etching with a strong acid, and then wash the acid out, and then 100% alcohol is to sterilize the whole thing up, and then flaming it just to remove all traces of alcohol, and then you have your cover slips ready. um Good question. So, yes, so that's a very good question. So, material is plastic, so potentially you can get some sort of a microplastic with it. So generally, these are used for short term. Okay, you're using your culture plates for a couple of months, and you're not heating it. You're not using, since they're growing cells, they're not going to be heated, or no harsh chemicals are often used. So people have honestly not studied this so much. What is the effect of uh, microplastics? Because the thing is, it's it's glass versus plastic. So there is not a great control that you have, which is microplastic, like plastic with microplastic and a plastic without microplastic, right? That's a wonderful question. I've honestly never thought about it. I'll see if I can find something, if I can find some study, but as far as I'm aware, a lot of biotechnology and related fields, they're heavily dependent on plastic, right? Pipette tips, epidophs, tubes, right? So, um, but I would assume the effect would be, like, the chance of there being microplastics would be minimal, because we are not using very harsh things. Acids and all, they're generally stored in glass bottles, right? So it should be minimal, but how much there is, I'm not sure. But good question. Yes, the bumps that are introduced upon etching, that is helping in the adherence of cell. And we'll talk a little bit more about this. But yes, that's the idea, why you want to etch your cover slips. So now, so that's the basic idea. So that's where the cell culture is done. And different cells, whether you're using stem cells, particular sort of cell, whether there is human cells or other cell types, other organisms, um, each cell has their own ECM requirements first of all, but also cells have some level of potential to generate their own ECMs. Now, but to be generating their ECM, they need to be adhered first. They cannot be floating and generating ECM. Not all cells can do that. Okay. Some cells can do that, not all cells. So for those, generally there is one or two components of ECM that they already want, just to be stuck to the base of your plate or your well or your cover slip, and then start the actual secretion. Okay, and that's what we are going to talk about now. So the ECM that cells need, the most important ECM that they need to just stick on, people have studied this. So there are a lot of things very, very cell type specific. And this is what we had briefly touched upon in, I think, last week, where we discussed what happens if you want to grow two, three different types of cells together. They would have their own requirements. So do you put a mixture of all those ECM components and make that first matrix for them to come on? In what ratio do you want that to happen? So all those optimizations need to be done before a protocol is published, that it says, okay, to grow X cell type, you need this in this concentration. For a Y cell type, you need this in this concentration. And if you want to grow it together, you want to co-culture it because you want to study the interaction of two cell types, right? You need to have, do you want like a different ECM altogether, or do you need a mixture of the first two ECM components? So that's the protocol making is already, it's one of the major things that happens in cell culture field. And then improvements on those protocols are equally important, because very often what would happen is, based on some ECM slash growth factor combination, what you may have is, you're seeing, okay, there is like 20% cell death, but that's the best you have currently. So you don't really have a choice. But then a protocol can come up and that can say, oh, we see better growth, or we see a faster growth. Okay, or we see, so generally when I'm talking about cell death, it's generally what is happening in the first 24 hours itself. So let's say you start off with 1 million cells, and what you see is, let's say only half of them have attached, right? So that's quite a loss. You need to start off with a big amount and then see what are attaching. Or over time, you can talk about the growth rate. Okay, today I have 50,000 cells, tomorrow I have 100,000 cells, or whatever. So what's the growth rate like? So anyway, so ECM components can be broadly divided into two categories. One is natural. So what are almost exactly as you see in your um in around the actual cells. So, and why I'm saying almost is because, for example, collagen. So collagen is a triple helix, and if you read about collagen fibers, so you might have, if not in the context of cell culture, you might have heard about collagen for a lot of skin creams, okay, cosmetic creams, because collagen is what makes your skin look nice. Okay? But then, of course, the type of collagen you have on your face, the skin on your face, versus what you have on your hands or your back or your legs, that's going to be different. Okay, because in a layman's term, you will say like your skin face is different from your skin on your hands. That's why you use a face wash versus a body wash, right? Broadly speaking. So collagen comes in different combinations, uh, or different ratios, different combinations and different ratios of those combinations. So what you see in an actual skin or anywhere else versus what we are using for a particular cell type in the lab, that can vary a little bit. And just an extra thing, that it's not dissolved in water, but it's in an acidic solution. We will not go too much into the chemistry of this. And we'll talk about, so collagen, like we talked about, you can have different combinations, and depending on that, you can have it as a film. And by film, we mean that general thing that we have talked about, the matrix that you have at the bottom of your well or the plate, versus we'll talk about when it's a gel. So again, okay, we can probably just talk about it here. So this is what was talked. So you want it to be like a thin film, which is just at the base of your um, plate, and your cells are growing on it. That's one thing. And that's very similar to what we just talked about everything else. So simple matrix just to adhere the cells. Versus in a gel, what you can have is that now it's no longer flat, but now it's cuboidal. So it has, so of course, this is also some thickness, but it's as good as 2D. And for gels, it's going to be 3D. So now it has some thickness. And depending on what sort of thickness you're talking about, cells may or may not go within the gel. So the cells can now grow inside. So you can put like a big blob of that thing. So it can look something like this. It can look like a dome. And then you can have your, let's say, you might have heard about spheroids. So which is a group of stem cells, and you just, or group of cancer cells that you just insert here. And then over time, they're just growing bigger and bigger and bigger inside that gel. Okay. So of course that, what sort of a porosity there is, what cell types are you growing, what's the size of the cells, whatever it is you're growing, do the cells, do you want to give the cells enough space to grow out of it? Are the cells going to have any projections? Things like that. So broadly, collagen, um, it can, there's a lot of things that can be done with collagen, a lot of studies. And since it has multiple types, again, multiple sort of ways in which it can be used as an ECM. So that's one thing. Um, then let's talk about laminin. So laminin probably is something you might have heard somewhere. So laminin, for whatever reason, again, we're not going too much into the reasoning, but laminin does not directly coat surfaces, whatever surface it is, whether it's glass or plastic. Laminin doesn't directly go very well. So PLO, which is polyl-ornithine, uh, and we'll just come to that. So that is coated first. So let's say you need to do like an overnight coating, or maybe like a 6 hour or 3 hour, whatever the, again, different protocols. So first, polyl-ornithine is coated. So you take your plate or your dish or cover slip, whatever it is, coat with PLO. Maybe there's like a middle wash step that you need to do to remove any excess PLO, whatever is not adhered. And then you add laminin, again, incubate for some time. And I don't know if, I don't remember if there's a wash step after it or not. Irrespective of that, then you can put in your cells, whatever the cells are that need laminin to grow on. For example, neurons, the cells I was talking about. So I do this step. So PLO and then laminin, and then do my neurons come on it. Okay. Gelatin, again, not talked about in too much detail, but that results from hydrolysis, partial hydrolysis of collagen. So collagen molecules, they interact with each other with some covalent bonds as well as with some hydrogen bonding. So when a partial hydrolysis is done, when some of those bonds are broken, so of course the hydrogen bonds are broken, but so collagen is mainly like a protein, right? So proteins are made of amino acids, and depending on what definition you go by, but there's something around 20 amino acids that we generally talk about. So, um, so this is, for example, one of the amino acids. So amino acids are mainly carbon backbone with some functional groups here and there. So, I think L amino acids are mostly what our body can make. If it makes the L ones, the D ones not so much, if I remember correctly. But anyway, yeah, I think that's what this is. So a protein is made of multiple amino acids. So for example, if I remember the figure correctly, so let's say 110 Dalton is what is roughly an average weight of an amino acid. Okay. Let's let's go with the number 100 for now. So Dalton is the unit in which we talk about weights of proteins. It's not grams or anything, but Daltons. So if you have two amino acids joined together, we'll broadly call it has 200 Dalton in size. If it is 10, it's going to be 1,000 Dalton or 1 kilodalton in size, and so on. Okay, so proteins can be anywhere between, again, big numbers, but like 35 kilodalton and so on. So 35,000 Daltons, right? So big numbers. So if you hydrolyze means remove. So they're all going to be, okay, so maybe I'm a little bit all over the place, but imagine that this OH and this H, they get removed. Okay, so this, sorry. Oops. Yeah. So this OH and one of the H here, that gets removed. So you have water that's gone out, and now this thing and this NH get connected. That's how you make these polymers. So basically, that's how proteins are made. Now, if you have a protein, if you have that polymer, big amino acid polymer, and if you do a complete hydrolysis, so every single link is cut when you get amino acids. But if it's a partial hydrolysis, so you might get some amino acids and you might get some chains, which are not the entire protein, but maybe a group of 5 amino acids together, maybe a group of 20 together. So those are called peptides. So whatever that is formed when you do a partial hydrolysis of collagen, what you get is gelatin. Okay. And you would have definitely, or even if you have not, so there's a lot of these, uh, I don't like gummy bears, if you've heard of those sweet things to eat. They're very chewy. So oftentimes, so in India, gelatin is not used, but a lot of places outside, if you see anything that's a very stiff jelly, it's not very wiggly. But if you see that, then you'll see in the ingredients gelatin is used, and gelatin is an animal product, typically derived from bones, bone marrow, and other sources. So a lot of people in India won't eat it. So, but yeah, anyway, extra information. So just remember that that's the sort of consistency it has. So you add gelatin, it can come as a powder. You can always like freeze dry it, and then that comes in a powder, and that is what you can add to things to make them. It's a stiffer form of jelly, but yeah, that's what gelatin is. So you can definitely make a gel out of it. Then there is another ECM component which is called concanavalin. Sorry, just give me a second. Yes, so which is called concanavalin A. Oh, I missed a term here. So it is a lectin. I'll write it better. So it's a lectin. Now, lectin is, so again, it is again a protein, but it doesn't bind to another protein, but it binds to carbohydrates. Just carbohydrates, or so a protein is when, so a molecule is called a protein when it's made of amino acids only. If a molecule is made of amino acids as well as some carbohydrates, then it's called a glycoprotein. Okay. So proteins or glycoproteins, we broadly put them in the criteria of proteins. Now, a lectin is also a protein which binds to only sugars or carbohydrates, or it can bind to the carbohydrate part or the sugar part of that glycoprotein. Cool. So generally you can have like membrane receptors. So a cell will interact with other cells through their cell membranes. So a cell membrane is lipid, but there are a lot of proteins or glycoproteins that are there across the membrane. So it will have a part outside the membrane, it will have a part in the membrane, and it will have a part inside the membrane. So the outside part interacts with other things outside, whether that's the ECM or whether that's the neighboring cell. It gets signals. Generally, what happens is it changes some configuration when outside is bound, and inside there is some other functioning or some other signaling molecule, which then drives the cascade towards the nucleus and things change broadly. So some proteins may just be proteins, like they only have amino acids, or sometimes they may have an added molecule, which is added later after protein synthesis, which is a carbohydrate. So why is a carbohydrate added to a protein, right? What purpose is it serving? Just to make it more modular, just to add more control, fine tune some things. So there are things which can identify that carbohydrate part, and those are called lectins. So concanavalin A is one such protein. So now you can imagine out in the ECM, you have concanavalin A. There is a glycoprotein on the cell membrane, and that glyco part most likely is going to be outside the cell. And that glyco part interacts with concanavalin A, and it's like, okay, yes, I would like to stick here, right? That's how your cell can get adhered to your ECM. So those were your natural things. And by direct or biotech based, it's just that what is your source? Are you deriving it from actual cells, For example, there is one name which was not, which did not come up here. That's called Metrigel. So again, that's what I use for culturing stem cells. So Metrigel comes from mouse sarcoma cell, like what that's secreted by mouse, some sort of a sarcoma cells, right? So the company that's selling these, they have mouse carcinoma cells uh, which release this, and that's purified and sold. Versus, are you doing it through, are you synthesizing it? Can you synthesize it outside of lab, or can you, again, we touched upon it very briefly in the first second class, where you can also use bacterial cells or something else to make these proteins in a large scale. So what sort of a setup you have, but ultimately the composition is either 100% or most likely what you see in the natural setting. Versus synthetic is what, they are a different form of what you see in the actual setting. They're not available in the wild, so as to say, but they can serve a good purpose. They help in cells growing, and most likely they would be cheaper to produce. So one of the things is poly D lysine, which we will talk about a little bit more extensively. So but yeah, the D here stands for these. So there are enantiomers of amino acids. So as you can see here, this NH2 group that's there in lysine. So lysine has 1, 2, 3, 4, 5, 6, or 5 CH2 groups. And it has this COH and NH amine and yeah, carboxylic acid. So that's there in all amino acids, by the way. This NH2 and COH is what you will see in all amino acids. And then you have a basic carbon-hydrogen chain backbone, and you'll have some functional groups. So lysine has this NH2 group right next to the carboxylic acid. So just remember this structure. So where this functional group is projected, it can have multiple projections. So based on where it is, it can be categorized as D or L. And like I said, D amino acids are generally what, so not all amino acids can be produced in the body. So some amino acids you need to take with food. So your food has proteins, right? So that protein is metabolized in your body, chopped up into amino acids, and those amino acids are directly used for other things. While some amino acids can be synthesized by the body. Overall, there are broadly 20 amino acids. But for example, ornithine is not an amino acid that is produced by the body. Lysine is very much there in the, like, not about produced or not. We don't talk about ornithine at all. Lysine, poly L lysine is one of the amino acids that we require. But D, on the other hand, is not there. But D is what is made in the lab. So now, depending on, so again, like for example, we talked about collagen. You can have it in different proportions, mainly talking about different concentrations. Something similar to that is what you can do with poly D lysine also. You don't want the gel, so let's just talk about the consistency. So you don't want the gel to be too thick, so it's going to be very, very high concentration. You also don't want the gel to be too flimsy. So it's going to be somewhere in the middle is what you would ideally want. But there comes another thing. So poly D lysine, depending on what the concentration is, its hydrophilicity also changes. So when you have low concentration, it's more hydrophilic, so more water loving, and high concentration. So if we just make like if we have concentration here on the x-axis, and so generally you can use both terms, hydrophobicity and philicity, but we'll just talk about philicity. Okay, so that's hydro, so water loving capacity. So for low concentration, it's higher, and for high concentration, it's lower. So this is the sort of thing you expect. And now of course, cells, they themselves are not water, but the inside and the outside of cells is all water. Even in your body, the inside of cells, the cytoplasm, It's mainly an aqueous solution. It's the base is water. There are thousands of things inside: salts, sugars, proteins, all other lot of biomolecules inside. The outside also is there, but the basic thing still stays that it's water. Okay, so if you remember, for those of you who might have studied about the cell membrane, the lipid bilayer. Why is it a bilayer? The lipid also has a hydrophobic and a hydrophilic end. So on both sides, you have the hydrophilic uh, portion that's exposed, both through the outside and the inside of the cell, right? So just remember from that. So cells would also want to be in a place because the things exposed on either end is water based. So you want to be in this hydrophobic zone somewhere, right? This zone is where you are aiming for this sort of a concentration. And yes, so that's dissolved in water. And in terms of collagen, so it's dissolved in acidic solution, but then things can be washed away, and then whatever stays on that cells can be grown. But it would still be, even if it's an acidic solution, it's still water, right? It's not organic solvent. We are still talking about water here. So you might, so if you read about this somewhere, um, you'll often see that there's an HBr that's coming up. So borate that's coming up in how this is available. But again, that's too much chemistry for this course, so we won't be going there. Datasilane, this was not discussed at all. It's just a name for your reference. Polyl or ornithine, we talked about this in terms of laminin. So just remember that laminin is a natural ECM component, but polyl ornithine, which is sort of a prerequisite for laminin coating, that is a synthetic one. So that's that. And I think this is one of the papers that that Menaksa was talking about in the lecture, that concanaval and how this came up. This is a very old paper, so it's 1985 that came up. That came up as an important ECM component to grow rods and cones, rod cells and cone cells, which are light and color detecting cells of the eye. So that's a lectin. Again, remember concanavalin is a lectin binding to carbohydrates, and that mostly came up in terms of being used in ECM for cone and rod cells. All right. So good so far with this slide. Okay. So I think that was the main crux of what was there. So you need a different number of ECM components. Okay. For example, in terms of laminin and ornithine, it was one after the other. Now, when protocols were made, so now mostly, for example, if some of you go to a cell culture lab for the first time, most likely you will have a protocol given to you that, okay, take, you know, coat the wells with 0.1 mg per ml of some, let's say, laminin for 5 hours, wash with water or wash with acid or wash with ethanol or wash with something. And then, or maybe if the first thing was ornithine, incubate, wash, then laminin, incubate, wash, then add, let's say, whatever. So oftentimes you'll be asked, okay, if you're using so depending on what culture grade you are adding, you want 10,000 cells per centimeter square to be added to it. So you will calculate, okay, if I have 25 centimeter square and this is so many cells per centimeter square, how many cells do I need? In the time it's being incubated, you have to do these calculations. You'll see where you are bringing your cells from. You'll count your cells, and let's say that's the protocol. But then, if you've had certain amount of experience, and now you realize, okay, protocol I'm using it's not good. Um, it takes too long to uh, grow these cells, or most of my cells are dying. Or if you're working with a rarer cell type, so you're like, okay, I don't see any papers that talk about a good ECM for this. I've done some work. I've analyzed some tissue samples and I've seen what ECM components there are. Now I want to make a new ECM component, or use a combination of existing ECM components and make a new protocol. So the first time that anything is done, you need to do extensive amount of characterizations so that that study becomes reproducible. If you come up with a protocol, I need to know what concentration, what temperatures, what amounts you worked with so that I can also do it. And then you have to give rationale that why is something working. You can't be trying 100 different things and say, okay, this is what worked best. So maybe you do that. But then for a paper, for a published work, you need to give a rationale. Why do you think that's working? It cannot just be empirical and only we tried things and it's working. It's great, it's working, but it's good to know why so that others can also build on it. So the why comes from some characterization studies. Um, so like one of you pointed out, those bumps that we are adding in the glass slide, you don't necessarily need to edge the surface as long as what you're coating it brings those bumps. Okay. Again, adding it to the cell culture dish or whatever it is, that helps. But then it also needs to be coming up in your the coating that you're doing. But then the coating does not have to be too bumpy also. It needs to be relatively flat. So that's sort of a surface visualization and just looking at the physical properties, that how many bumps are there in a certain amount of area, what is the height or the depth of those bumps. Okay? Those characterizations are done with something called the atomic force microscopy. Um, and this is not just a technique that's used by cell culture people. In fact, it's very rarely used by cell culture people. This is more into, I think, material sciences. People could be synthesizing their own materials and then you want to characterize it. Okay, these are the properties. So this setup, what we are here seeing in this slide, is more into that field. But yeah, but then it's also used whenever you're making something new. So that's one thing you want to do, just looking at it at a microscopic level. That's what AFM does. Then this is where we talked about the hydrophobic and hydrophilic calculations. So for example, when people realize that what sort of a concentration, you know, suppose you were also making a new synthetic PCM component and you want to check what sort of a concentration would work best. So you try 5 different concentrations, okay? And then you see what is the trend with the hydrophobic and hydrophilic or hydrophilicity of that thing. So in this case, what you can see here, so this is an image from AF, by the way. So you're taking a square area that you can see. So that's the X and Y dimension. And depending on what color you're seeing, so it's going to be a black and white image. So depending on what color you're seeing here, that's, I think that's either the height or the depth. I'm not sure. I've not worked with this AFM, but it has something to do with in the Z-axis. If any of you know this, let me know. But otherwise, the color intensity or the color here is telling you what difference you're talking about as compared to a base. And then for contact angle measurement, just telling you. So what is basically happening here is, so you have a surface here, and then you've coated it with some concentration of your material. Okay? So let's say here we are talking about 3 different materials or three different concentrations, and you put on a drop of water. So we're talking about water lovingness here. So put on a drop of water, and what the machine, which are called goniometers or tensiometers, what they're doing is they're just looking at the shape, quite literally. And so what you can see here, the flatter it is. So again, just imagine what is the amount of area that's covered. So this is the area. Here it's a shorter area that's there, and here it's an even shorter area, right? So imagine this is almost the diameter of the cell, of the sphere or the circle here. This is a little shorter, then this is like very, very short, right? So how flat it is tells you how much surface area your drop is covering. So that's one thing. Or this can also tell you you can also measure the same thing depending if you calculate what the angle is, if you measure what the angle is. So in case of this flat drop, you have a smaller angle that's made. So a contact angle, which is short, so a flatter thing, that indicates a hydrophilic material. If the angle is more than 90, if it's more than 90, then you call it a hydrophobic material. Again, actually, what I said about the base length is actually not true, because what if it was even flatter? So you can ignore what I just said. Yeah, this thing is not 100% correct. But yeah, but the angle is a, for sure, it's a better measure. So the contact angle, if it's greater than 90, that's hydrophobic. And if it's even bigger, so the maximum it can be is 180, right? But if it's even bigger, then we just call it super hydrophobic. So, and the drop that you place, it can probably just slide off. So this example was given that if you, so leaves, for example, they have some sort of a waxy coating on top of them so that they don't lose a lot of water through evaporation, right? So you might come across, especially in winter mornings, there's dew that's accumulated on leaves, right? So this is what's happening. The drop is almost a perfect sphere. It has a very small um, part of it that's touching. So the contact angle is really large. So it's a hydrophobic. So the material is hydrophobic, right? So that's one thing. Now here in case of AFM, we did not know what's there on the surface. We just knew what it looks like, physically what it looks like. And then if you want to identify, so again, like we said, for example, for lysine, that's a relatively simple molecule. But now, what part of lysine is exposed? Is it this NH2 that's exposed? Is it NH3 plus ion that's there? Or does it interact with something in the media and then it becomes a new molecule altogether? Or, for example, if you were working with collagen or gelatin, what's the proportion of different materials that are exposed? So atoms or molecules or functional groups, how much are they? So that chemical analysis. So again, this was just a physical analysis of the surface, and now you're going to do a chemical analysis of the surface. But the fact still stays the same, that you are talking just about the surface. So, so what you want to be doing, and this is not just what there is, and it's also about how much there is, right? Because if you're testing different combinations, for example, you want to test what happens if I'll add poly D lysine and collagen, or poly D lysine and, let's say, beta-silane together. What concentration do I add them as? Suppose I added them as like 25-75, 50-50, and 75-25. Let's say I'm adding them this ratio, and now I'm putting them in different things. And I saw let's say the surface looks pretty much the same. The shape, the AFM graphs, they look roughly the same. Your contact angles are the same, but then where's the difference? So maybe the difference comes up here, or maybe the contact angle is also different, and then the difference also comes up here. Or what can happen is maybe one of the two things is heavier, maybe one thing settles. So always you have the same thing that's there on the top. So ultimately your cells are only looking at what's there at the surface. Surface chemical analysis is important. We'll talk a little bit about if you want to compare what's there below the surface. But yeah, for surface chemical analysis, so what you have is you have a graph or a reference, something like this. Again, I'm not very well equipped at what this is, but for example, the x-axis, so mainly the x-axis, whatever the value is in this reference, each value is pointing to a particular atom. So for example, at 300, you see there is a peak which corresponds to C. At something around 500, there is a peak which corresponds to molybdenum. At a little higher, there's a peak which corresponds to oxygen. Somewhere here, there's zinc, nitrogen, different um, different states of zinc. Different different things are there. Now, what you do is, suppose this is one side, and then what you do is um, you do the analysis throughout the spectrum. Okay, you cover between 0 to, let's say, 1500, or however much that is, or let's say it's up till some a little over 1000 here. So you do that, and then you're zooming into So you already know this. Now you got some peak at, let's say 1020 or 1022, let's say this point. Okay. And then you go back to this reference graph and you see, okay, most likely this corresponds to some uh, valency of zinc. Okay. And then, of course, the height of the graph can give you an indication of how much that intensity can tell you if it's more, if it's less, right? So something like that. Of course, this sort of you might sometimes have to do some deconvolution. For example, in this graph, you see one giant peak. Okay, this inside color is not what you get from the graph. This is what you have identified. You get this shape. So you see, okay, I can see that there's a peak at a little less than 162. So let's say you see a lot of something here. Okay. Let's say you see something like sulfur here. But then there are tools and algorithms which can say, is it best when you fit a single molecule here or a single atom here? Or can it be better if you fit two things together? So if you get the actual shape of the graph and fit in two things, then you're like, okay, there are two atoms which are exposed to the surface. So briefly, that is what. So this is X-ray photoelectron spectroscopy, XPS. That's what you do when you want to see what is there at the surface and how much of it is there. All right. So good so far. Are we all on the same page? Yeah. Let me know if there's any confusion. So now this sort of thing needs to be done when you're doing something new, not when you already have protocol in hand. So that's that. Okay. So a little bit more about collagen, especially when we're talking about collagen as a gel. So collagen needs to be, if it's in a gel, you want to, so depending on how much it's cross-linking, how many of those more covalent bonds is one collagen triple helix making with another collagen triple helix, that is going to decide how rigid the gel is and what sort of distances are there. Um, so you need to have some empty spaces. It cannot be so collagen, it's a molecule, right? And you most likely want to grow cells. So cells are bigger than collagen, right? Imagine it's like there's a spider web and you want to fit I don't know, like a football in that web, in that spider web without breaking it. So you do want there to be spaces in the spider web to fit that football. Okay. That's a very not the best example, but I hope this visualization helps. Okay. So whatever those connecting molecules are, so we'll just call them as handler molecules. They can be collagen or they can be other peptides. They can be polymer. Again, that's a whole different field. So we'll not go too much into it. So depending on what your um, function, like what your task is, what's your main application, you need to identify what porosity are you talking about, what size of the pore are you talking about, right? So that needs to be calculated, and that's depending on what you want. You may also want to put like a smaller thing inside it, some other material, okay? Nanomaterial, for example. Okay. So it depends on what your function is. So what should be the porosity? And you don't want the cells to be too tightly. Now, if a cell is going inside that mesh, you don't want that the gaseous exchange or the diffusion should be affected, because finally, yes, sure. So, um, so imagine collagen is like a triple helix. Okay. Now, the broad idea is that suppose you have a big mesh, like a 3D mesh. For example, it's a cuboid that's made just of spider web. Okay. So spider web can have very intricate designs, but we generally look at spider webs in 2D. But imagine it's in a 3D. Let's say one room is entirely full of spider webs. And they're strong enough to hold like some sort of a football or some sort of a ball in them. But you don't want to break the spider web in that process. So the web needs to have um, enough spaces between them. So now in this case, when we're talking about the web, the entire thing is one material. It's all, for example, it's all collagen. But you might in the lab when you're synthesizing something like a web, you may not just have collagen, but you might have some other things that's connecting. So a spider web can have like major things, and then there can be finer web that's connecting the different things. For example, so just imagine if you have like another helix here, what's connecting these two things? Now you want enough space. If they were close to each other, they would just interact with these molecules that we talked about. For example, this NH2 can interact with something else. Their collagen has a lot of amino acids, so there are a lot of functional groups. So there could be something here throughout. If they were close, this can just interact with this directly. But we want them to be far apart. So we want something to connect them. That connecting thing, it may be another collagen molecule, it may be some other peptide, which is like a shorter segment, but it's just a set of amino acids, or it could be some other polymer itself, right? Now that can add, so depending on what length you're talking about, what configuration it's, you can have different pore size. So this is your pore size where that football or whatever can be filled. And this can also be big enough to fit cells. Yeah. So that's the handler molecule we're talking about. What or handle molecule, maybe handle is a better term. But what is connecting your collagen molecules and contributing to the pore size? So porosity is mainly pore size and how many are there. Okay. So, but you don't want this to be too tight also or too thick, but it should be enough to allow nutrients and media to go in out, gaseous exchange, everything's happening through diffusion. So that should not be affected. But yeah. So now, now what is happening here? Again, I'll just draw out one more thing. So, for example, if this was the base of your culture plate, what you're doing is you are making a, let's say this is a thick dome. And now, question? No? All good. Fine. So you're making this big dome out of collagen. Okay, so this is uh, collagen. And what you're doing is, let's say you are adding a group of cells inside this, okay? So 1, 2, 3. I'm just drawing a few here. So this is what you're growing. And then they're going to be happy there, then they're going to start growing. Okay. So so yeah. So this is how the setup is going to be. Or it may have just been, it could be also be like this, that this is how you're growing it. Right? It can just be like a gel that's covering the entire base. If you want to grow something 2D on this, relatively 2D, so you can also have your cells growing here, but now they're covered by this collagen thing all throughout. Earlier, what we were talking about is you have this, whatever is the coating here, and your cells are growing on top of this. Like they're connected, like they are touching it, but they are just exposed to this at the base. But now your cells experience this gel throughout them. Okay. Now, what's the use where this comes into the picture is going to something we'll talk about later. But yeah, let's say this is what you want to do. So until now, we were just interested in the surface because that is what was exposed to the cells. But now the cells are experiencing the whole of it, right? So you do want to check that as you cut across. So imagine you know, if this is how it is the collagen part here or here or here, is it the same all throughout? Yeah. So for example, your entire body, for example, that's where your cells are. You're not the same here as you are everywhere else, right? So you want to cut through and see, is there something happening because of density? Like, is the density changing? Are some parts settling? So is the bottom of your gel different from the top of the gel? So on. So what you can do is you need to cut through the gel, for which something like electron beam lithography can be done. So mainly you're cutting through, and then now you have a new surface that's exposed, right? You've cut through the whole thing. So now there's another thing that's exposed. So now surface analysis can be done with the same things here with this part. Then, you get a better idea of what your cells are experiencing. So that's one more step for characterization. And this is another thing that was just discussed in the class. So one source of collagen, again, where does collagen come from? So one source is a rat's tail, which is rich in collagen. So when a rat is sacrificed, so with a pair of clamps or plier-like devices, which are called hemostats, that's twisted, and the white threads that come up, they're collected. Those are dissolved in organic solvents, run through a chromatographic column to purify, to mainly extract that. Then HPLC is done to purify. So again, just an extra information, but yes, a rat tail is a good source of collagen fibers. But again, of course, what you get in that is one sort of a combination. Another source would have a different combination. So that's that. And I think this is the final thing. So once the steps, so we talked about characterization, right? ECM components, you characterize it. You may want to characterize by cutting it also. Once you do that, then you want to check what's happening to the cells. Yeah. Once you put your cells in, what sort of a contact angle do you see then? Because now cells are going to, so what I mean is add in your cells, you check what is happening to the attaching, like how soon are the cells attaching. They would generally take a few hours. They might settle. So what happens is, so imagine if you're adding um, sand to a water. So you have water in a glass and you're adding sand to it. It's going to settle in some time, right? Initially, like as soon as you add it, if you would swirl around the glass, of course the sand is going to go around. But then maybe after 30 minutes, if you move it again, they're not really, they're not attached, but they're not going to move because they've settled down. So that also applies to cells, because cells, they will settle down. They're big, right? So they will settle down. But have they really attached? So what you might want to do is tilt it and see, are they moving? Okay, the swirling may not, it does tell you, but then tilting is another thing. Are the cells moving now? And you can do this. You don't necessarily need a microscope for this. If you have a good enough concentration of cells, because they do make the base a little hazy. But yes, of course, if you have a microscope, just look at it, tilt it while you're looking at them under a microscope, and you'll see if things are moving. It doesn't have to be a big tilt, just a little bit, and you'll see things sliding. Okay, and then check the contact angle. Um, if you just, when the cells are attached, check the contact angle. And one more thing that's done is, so we discussed briefly here, we talked about it, that different cell types, they can generate some of their own ECM. What the cell types do with what ECM you've provided is that cells have some molecules that are exposed out from their cell membrane, and they can interact with the ECM that you've provided based on that. Now, cell type A and cell type B will have different of these molecules that are exposed, and they can interact with different things. That's why they need different ECM components. That's the thing, right? Now, uh, based on what sort of things they have, they may interact differently. So, for example, some cells, they may interact based on charge. So what you've added has a slight positive charge. What the cells have, let's say they have a negative charge. So they interact electrostatically, and then that is what attaches them. Sometimes they want to actually make covalent bonds. Okay, so yeah, so for example, PDL, poly-D lysine. So that is positively charged slightly. This NH2 that you see here, that takes up water. So that's NH3 plus now. So that's positively charged. So that's PDL. And cells which have a negative cell surface charge, that for them is what PDL1 is a good ECM component. So then cells will be like, okay, they've settled and they're like, okay, there's something to stick onto. We are happy. This feels home. So let's now furnish the home with our own ECM secretions. So negative cell surface charge, and they interact electrostatically, they will get enough time to stabilize and secrete their own ECM and adhere properly. Now, if it's something, depending on how strong the positive charge is, they may take more or less time. If you check after 30 minutes, what's happening? If you check after 1 hour, what's happening? If you check after 24 hours, how many cells have attached? So you can have like, again, take three different wells, three different, or nine different wells, 333 with different positively charged ECM components, and see what proportion of cells are attaching, how many cells are dying the next day, count again, things like that. Or with collagen and laminin, for both of these, cells have something called integrins on their surface. And integrin, different again, there are a lot of different subtypes of integrins. Something like collagen, some of those integrins like laminin, some integrins will like a particular combination of collagen and so on. But broadly, integrins can identify collagen and laminin on the ECM, and it's not charge based anymore, but they will directly interact like a ligand receptor interaction, and then that's how the cells get attached. All right. So that is what was covered this week. I hope I've not missed anything. And we are almost an hour and a half into this. So we'll have I just have like a handful of practice questions for you. These are a little bit similar to what you will have in your assignment. So just to give you an idea, I'll open the chat box here. Uh, yeah. So just pop in 1, 2, 3, 4 or A, B, C, D that you think is the right answer. So poly D lysine monomer has how many nitrogen atoms? Yeah, yes, it has two. Good, right? So just remember this structure. So this is the monomer, right? So this N is a part of the main thing. So yeah, so that's two. But yes, it's a slightly confusing thing, but so that's two here. Which of the following is a synthetic extracellular matrix? Gelatin, collagen, laminin, or polyl ornithine? So, yep, great. Okay, good. Others should also reply. It's okay if anybody is not, no problem. So, gelatin is a hydrolyzed form or a partially hydrolyzed form of what? So, we did not talk about elastin or fibronectin, but these are all also ECM components, different things that you'll see in ECM. Okay. Atomic force microscopy helps us analyze what? The chemical constituents of the bulk material, the topography of the surface, the concentration of the material, or the electrical stimulation of cells. That's the topography. So topography means shape, heights and widths and heights and depths of a surface. X-ray photoelectron spectroscopy, which is XPS, that is used to analyze what? Topography of surface or bulk, or chemical constituents of surface or bulk. Chemical constituent of the surface. So surface is what we're talking about. If you want to analyze the base or if the bulk, then it needs to be cut, and then that base can be used as a surface. So for example, if this is what you're doing, so this surface can be directly checked with X-ray photoelectron spectroscopy. But if you want to analyze the bulk, then what needs to be done is you need to cut this through, and then this is the surface that you want to do XPS on. So sort of you get an idea of the bulk, but what this technique is just for the surface. So yeah. So that's what this is. Um, contact angle contact angle on the surface is indicative of what and is measured using a what? So again, hydrophobicity is not a term that people generally use. I mean, it's either way, hydrophobicity, philicity is fine, but what the term is hydrophilicity. So again, it's a goniometer. It's there. Osmometer is something we talked about in the last week. We get the osmolarity. Concanavalent A is a what? So it's just a little wordplay here. So, so it is a protein that can bind to a carbohydrate. So ideally, there should be like a hyphen here. Just copied this in. So now, would you like to stick to your answer, or would you like to change it? So concanavalin A, is it a protein or is it a carbohydrate? Yeah. So it's a protein. Yes. And it can bind to a carbohydrate. So that's why this term is correct. Because this one indicates, I mean, what the way this would have been correct is protein binding to a carbohydrate. That's there. But mostly the way this is talked about is it's a carbohydrate that can, it's a protein that binds to carbohydrates. Can you just give me a second? I'm getting a call. Sorry about that. So yeah, so it's a protein that can bind to a carbohydrate. All right. And then final thing. So retina of the eye that contains rod and cone cells. Okay, we did not explicitly talk about this today, but rods and cones are to detect different parts of the light. One of them is for the intensity, and the other thing is for color. So it's just simple to remember this way. So I'll just label this. So C stands for cones and C stands for color. And the other thing is for intensity. That's how I remember it. So yeah, that's that. And that's about it. Um, for so after this, we can take a quick look at assignment 3 solutions. If there's anything you want to talk about that, I'll anyway just go through it quickly. I think there were just 10 questions. So yeah, the main concern when you're designing a cell culture lab is contamination controls. We talked in detail about where should things be placed. A dissection area and a cell culture area which is sterile, they should be kept separate. Mm, a laminar airflow or laminar flow hood that ensures sterile working conditions, and we talked about horizontal and vertical and all of that. Incubators in a cell culture lab are used to maintain cells at a particular optimum temperature and CO2. Autoclaves are used to sterilize equipments and media. Long-term cell storage, ideally it should be liquid nitrogen, but since liquid nitrogen is not an option here, um, it's -80. Uh, so I am not 100% sure of what the criteria is, but I think some best assignments are taken. Like I think it's 6. If anybody knows the exact number, they can correct me. So I think missing on one assignment should not be a problem. But you can take a look at the course page, and I'm sure somewhere I'll just come to it in a second. We'll just go through this and I'll let you know. Yeah. So yeah, so freezer type, it is good for long-term cell storage. Um, I mean, it says freezer, so liquid nitrogen is not really a freezer type. So the best thing that you have here is the coldest temperature, which is -80. Cryopreservation is done when you're talking about liquid nitrogen, that's 190, -196. Cells in vitro, they are as a single layer, and we talked about because that's the only in a single layer can the cells be exposed to that ECM that you just made. If it's going to be in 2, 3 different layers, then the top cell is has no longer any exposure to that ECM. Yeah. Glass or polycarbonate, um, that is used because it's transparent. course, because when you want to image cells, you want to image them from the bottom, so that's transparent. And they're easy to sterilize, and you also do get them already sterilized. And the interaction of the cells with the surface is important so that the cells can attach, they can spread, and they can grow. And yeah, so it's 8 out of 10 for the assignments, but I think it would just be 8 assignments for this course, right? So maybe it's going to be a little less, either 6 or 7. I think there should be, I'm not sure where it's come up. Would it come here? But yeah, you can just take a look at the um, assignment. I don't know where this comes up. Give me a second, I'll just go through. Hmm. Yeah, I'm not sure where that comes up. But yeah, you should find this somewhere. But yeah, it's something like that. So missing one should be okay, but yeah, just keep in mind to not miss any of the other ones. All right. So with that, we come to the end of today's session. This recording is going to be uploaded to YouTube right after this. Yeah, you're welcome. And I'll upload this PPT on the Google Drive. And again, you'll have the link for this here. So if you go on your uh, course page, you'll have this tab for a problem solving session, and you go to the recordings bit of it. And here it says click here, and you'll be redirected to a Google Sheet. And yeah, so this is the presentation, and this is the individual YouTube links. So click on that. You can watch if you want to just go back through any of the previous ones also. And the assignment for this week's assignment, the deadline is day after tomorrow. I think it's 11:59, but it's just 10 questions. So solve it as quickly as you can. And I will see you all again next week, Monday, 22nd September, same time, and the Zoom link also stays the same. I'll stop sharing now. Please let me know if there's any question that you may still have. Otherwise, that's about it. Thank you. Yeah, all good. Then you can leave the meeting. I'll end it in a minute. Thanks for joining, everybody. Thanks.